Okay, great. So our next speaker is Iris Farnum. Iris is a rising senior as well. Yes, I have to make sure I have all these right. She's majoring in biology and Spanish, and she spent the summer in Jim Gosella's lab. And Iris comes to us from Portland, Oregon. Iris.
Thank you. Hi, everybody. So again, my name is Iris. I'm going to be a senior this year at Eckerd College, and this summer I was working in the Gosella lab studying the effect of genetic variation on branaplam splice modulation. So the lab is mainly focused on Huntington's disease, which is a neurodegenerative disorder of the central nervous system. And HD is associated with the expansion of repeating CAG nucleotides in the DNA. And this repeat expansion causes the loss of vulnerable neuronal cells, which we can see in this top image. On the left is the normal brain, and then we see on the right the brain with Huntington's disease, which you can tell is a lot more atrophied, specifically in the striatum. And the average age of onset of HD is 30 to 50 years old. This summer, I was mainly focused on the therapeutic branaplam, which was initially developed for spinal muscular atrophy, which is characterized by low levels of the SMN protein. Branaplam works to correct the alternative splicing of the SMN2 gene in order to raise the levels of the SMN protein. So branaplam is a splicing modulator, and I was specifically focused on how branaplam includes a pseudoexon into a transcript. And in Huntington specifically, this pseudoexon actually lowers the levels of the Huntington transcript by introducing an early stop codon. Our hypothesis this summer was that genetic variants will affect the splicing modulation of branaplam. And something important to note is, although branaplam was in clinical trials, just on Monday, they announced that branaplam is being stopped in the dosing of the clinical trials because it seemed to be causing nervous system damage in the patients. And so it is, although this is really sad news, they can continue to look at the drug because it may be, its effect may be, the nervous system damage, may be because it lowers the levels of the mutant Huntington as well as the wild type Huntington transcript. So that's also something to keep in mind. So previously in the Gosella lab, variations of branaplam splicing across cell lines in the Huntington gene were observed. So in this graph, we can see on the y-axis is the proportion of the normal exon. And then on the x-axis, these are all of the cell lines that were tested. And all of these cell lines are patient-derived, and they were chosen because they had a variant of interest. So you can see in some of these cell lines, like these first two, the normal exon proportions are very low. However, in some cell lines, such as the one right in the middle, there are high levels of the normal proportion, of the normal exon proportion. And so that just means that less of the pseudoexon is getting induced. And so branaplam is not as effective in that cell line. This summer, I was focused on other genetic targets of branaplam. So we first identified these targets, and then I developed new primers. We ran concentration gradients to find the best branaplam concentration. And then we ended up dosing the cells with 100 nanomolar branaplam. I then ran PCR tests and then used gel electrophoresis to visualize the results. And finally, we quantified them using ImageJ. So all of these predictions were made using Splice AI, which is a deep learning tool that just predicts the effect of a variant on splicing. And they're all made by Daddy Gao. So Daddy identified the other genetic targets of branaplam from recent publications and then identified those sequences. Next, he found all of the possible variants of the genes and then predicted the effect of the variant on splicing using Splice AI. Then he found the allele frequency, and finally we went through our large catalog of patient-derived cells to see if we had cells with the variants of interest. So after all of that, we identified two genes in which we had cell lines with the variants and that had a high enough Splice AI score. And the Splice AI score is just the probability that the variant will have an effect on splicing. So after we identified these two genes, I designed primers, and we can see in this image, the primers read across the splice junction, and then when the pseudoexon is included, it will read over the pseudoexon. Next, I ran a concentration gradient to determine the best branaplam concentration. And for both TENT2 and ZFP82, we found that 100 nanomolar branaplam was the best concentration. And that's just because the levels are right in the middle, so they can go up or down depending on the effect of the variant. So after we dosed the cells, and then I started to run PCR tests, and then we used gel electrophoresis to visualize the results. And in this image on the left, we can see in the second lane, that was our control. The cells were just treated with DMSO. And here we can see that there's just one band around 0.1 kb, and that is the normal exon. Then after the dosage of 100 nanomolar branaplam, we can see a second band around 0.2 kb, and so that is actually the pseudoexon that was included. Then using ImageJ, we graph these results. And again, on the top is just the DMSO. There's one peak for the normal exon. And then right below it is the 100 nanomolar branaplam. There's two peaks. One is the normal exon, and then one is the pseudoexon, which you can see is a little bit more intense than the normal exon proportion. So I'm first just going to discuss our results with the ZFP82 gene. In this graph, again, on the y-axis, that is the normal exon proportion. And then on the x-axis are the cell lines that we've had. And again, they're all patient-derived, and we chose them because they had the variants. So you can see the first 5 are pretty similar across the cell lines. There's not a huge difference. But then when we get to the last cell line, we can see that it's slightly elevated. So again, that just means that less of the pseudoexon is getting included. Here in this graph on the right, we can see this is the actual SNP for in the cell lines. So when it's homozygous and it does not have the SNP, that's when we see the lower levels of the normal exon. And when it's heterozygous for the SNP, then that's when we see the elevated normal exon proportions. And so this just tells us that this variant may have an effect on splicing, but this is the p-value that we got from our t-tests. Now for the TENT2 results, we can see that they have much more dramatic results. First, in the first cell lines and then the last cell line, you can see that they have the lower normal exon proportion. But the 4 in the middle, they are much higher. So again, less of the pseudoexon is getting included in those cell lines. This table just shows all of the variants that exist in the gene, and the one that is boxed in gray, that is the only one that was predicted by Splice AI to have an effect on splicing. So although there was just that one that was actually predicted by Splice AI, we can see that these 4 other SNPs seem to be associated with the variant as well. And this can be likely due to linkage disequilibrium, which just means that the variants, their physical location, make them more likely to be inherited together. So again, it's really interesting. You can see when it's homozygous and it does not have the SNP, that's when there are lower levels of the normal exon. And when it's heterozygous, it has the variant, that's when we see the higher levels of the normal exon. We then decided to test TENT2 on the cell lines that were actually used for the Huntington gene testing. And so all of these cells were dosed with 50 nanomolar branaplam rather than 100 nanomolar. And they could just kind of confirm our results, where we see some are pretty even throughout and they're lower, but then certain cell lines have the higher level of the normal exon proportion. Again, on this graph, we can see the SNPs that exist in the gene. The one in gray is the one that was predicted, and then the other 2 seem to be associated with the variant, but again, are probably in linkage disequilibrium with the predicted variant. We can come to some pretty interesting conclusions from these results. The first is that genetic variants can affect branaplam splice modulation, and that's just important to keep in mind. These drug companies are testing them in patients, and so patients may have a different reaction depending on the variants that exist in their genes. Second, it's really interesting how Splice AI was able to identify the haplotypes in ZFP82 and TENT2, which cause branaplam to be less effective. We can continue to use Splice AI as a deep learning tool to predict the effect of splicing or of variants on splicing. But this also leads to the question of which allele is causing the variation, because again, there were a lot that were associated with the variation, but we can't assume that it was the one that was actually predicted, but that is just an interesting question as well. The next steps, the first is to sequence TENT2. All of the SNPs that I had were all actually imputated, so they're all estimated. So we're actually working on the sequencing right now. Hopefully we get our results in the next day or two so I can see them before I leave. And then another mechanism of action of branaplam is that it induces an alternative 5' splice site, which we can see right here. And so it'd be interesting to test how the variants affect that splicing. I'd like to thank Jim and Zach for working with me this summer and Daddy Gal for all of the predictions. And thank you to everyone else who helped me this summer.
Questions for Iris? Questions? Okay, thanks, Iris.